First of all, I would like to thank Esper for the invitation. It's always a great pleasure to come, to Brazil. And then, I accepted with a lot of enthusiasm this invitation. Now, I'm going to talk today about some, a project that we have in our, institute, which is not yet published. The data are not published yet. And then it's regarding, the concept of functional avidity and TCR renewal in HIV infection. As you will see, I will focus most of the talk on the functional avidity, then to understand what is the impact of having HIV-specific CD8 T cells with different level of avidity and the control of virus replication. Now, here are summarized some of the most important functions which have been recognized, mediated by T cells. Then T cells can secrete cytokines. There is a variety of cytokines, and different patterns of cytokines have been associated with better or less control of virus replication. Proliferation capacity of T cell is very important because it's one of the most consistent markers of control of virus replication. If your T cells proliferate well in response to antigen stimulation, then there is better control. Then the ability to kill virus-infected cells, also very important. And then there are, two other parameters which are very important because indeed the breadth of the immune response or the avidity of the T cells for the antigen are going to have a substantial impact on the capacity of the T cells to mediate the immune response. And then we are going to concentrate then on understanding why, what is really driving this different level of avidity in HIV infection. Now, to give you some background about avidity, for example, in the cancer field, it has very clearly shown that if you have cells with higher avidity, you are able to better eliminate your cancer. It has also been clearly shown that if during an acute virus infection you have cells with higher avidity, then you are able to more efficiently clear the virus. Then there are several publications which have shown, although I think that the data in the case of HIV infection are more controversial, but there are some publications showing that high avidity is associated with better control of virus replication. In contrast, we have lower avidity has been associated in cytomegalovirus or Epstein-Barr virus-specific T cells, and also some papers have shown that HIV-infected people have cells with lower avidity. Anyway, something that you need to keep in mind in what we are going to say, that what it has also been shown, which is very important, is that if you have a T cells, CD8 T cells, which has high avidity, then what is going to happen is that it's going to cause mutation in the virus, because more higher is the avidity, more the immune pressure is on the virus, and then the virus as a result of this pressure is going to mutate in order to escape the immune response. Then paradoxically, you may have high avidity cells, they induce virus escape. Now, then how we measure functional avidity? Because it's very important to first understand, in order to interpret the data. Then what I'm going to show you is that functional avidity, it is measured by the capacity of T cells to respond to a specific antigen under experimental condition of a decreasing antigen concentration. And then this is a classical example. As you see here, you have decreasing concentration of antigen. Here you are measuring the percentage of HIV-specific CD8 T cells able to produce gamma interferon, and then we measure this by flow cytometry. And then you can clearly see here that depending on the concentration, you are able to induce different percentage of interferon gamma secreting CD8 T cells. And then what we define as EZ50 is the antigen concentration that is going to stimulate 50% of the total of HIV-specific CD8 T cells. Then what we are going to measure in the rest of my talk, I'm going to express the data as EZ50. And then just to add, here are two examples of two, antigen-specific immune response. The CD8 T cells from these patients, they recognize two different epitopes, one restricted by HLA-11 and one is restricted by HLA-D8. And then these two responses have a very different avidity. And then generally, if you have low avidity, you have a high EC50, which means that you need much more antigen in order to stimulate 50% of the total response. In contrast, if you have high avidity, you have low EC50 because you need much less antigen to induce 50% of the CD8 response. Now, in order, most of the publication in the past, they have looked to few patients and then to patient, either primary infection or patient in chronic infection. Then there has never been really a systematic, studies in order to understand how this avidity may evolve within the same patient, within the same response over time. And we have studied three cohorts of patients. One is a cohort of patients of primary HIV infection. We are extremely efficient, I have to say, in Lausanne in recognizing primary infection. It's because I think of the healthcare system, which is very efficient. And then most of since we have in my division of immunology allergy, we perform screening for the P24 for the antibody, the HIV screening assay. Then we have the possibility to identify very rapidly, a patient with acute infection. And then, in fact, in this cohort, you can see here the average, viremia in this patient with primary infection was almost 8 logs at the time of the first. Then it's an average of 27 million copies per milliliter of plasma. And then, indeed, we have generally very well recognized what was the time, and then generally most of the patients, they come to our attention 2 weeks since the onset of the symptoms. Then we get this patient very early, they're all seronegative, and then the other advantage is that generally we initiate therapy within 24 hours. Then, we put, this patient on therapy very rapidly. Then this is the cohort of primary infection, and then we have compared with the other two cohorts, patients with chronic infection that we have studied prior initiation of therapy, following therapy, and then a cohort of long-term non-progressors that are people that with stable disease with HIV RNA copies less than 1,000. So then in order to study characterize the CD8 T cell response, we have used a pool of 194 optimal HIV-derived CD8 T cell epitopes. And as you can see here, during primary infection, for example, a chronic infection, then we see this is the distribution of the responses that we have observed against different protein of HIV, and then this is the frequency of total HIV-specific CD8 T cells. And there is not really much difference in the frequency between primary and chronic infection. Now let's start now to try to understand, first of all, what are the differences from a differentiation stage between a primary and chronic infection, and then we are going to move to understand how these then differences may correlate with Avidity. And then here in these slides, then we have looked at 3 markers which are able to differentiate, to identify very well the stage of the differentiation, CD45 array, the IL-7 receptor CD127, and the CCR7. And then as you can see, there is not much different. Most of the CD8 T cells in patients with primary or chronic infection have an effector differentiation stage. Then they are mostly negative for CCR7, CD127, and CD45, the typical effector phenotype. Now, if you look at co-stimulatory molecules, is another way to look at the differentiation stage. You can see that the patient with chronic infection, they have much less CD27, CD28, which again is consistent with the fact that there is an effector phenotype and there are differences. But what is really important are some differences in the functional profile between primary and CD8 T cells from people with primary infection and people with chronic infection. In fact, as you know, probably, here we are looking at gamma interferon, IL-2, and perforin. Now, in a patient with chronic infection, this is prior treatment, then you can see, as it is very well known, the patient are mostly, have CD8 T cells, they mostly secrete only interferon gamma, then they have a monofunctional profile, and they are largely defective in perforin. There is almost no perforin expression. Perforin is very important because if you don't express perforin, you are not able to mediate cytotoxic activity. But interesting, if you look at patients with primary infection at baseline, prior to the initiation of treatment, you can clearly see that about 50% of these patients, in contrast, they express perforin. Then these cells at during primary infection potentially are able to kill efficiently virus infected cells, which is in contrast not the case later on when the patient progresses to chronic infection. And then they have therefore a more polyfunctional phenotype as compared to patients with a chronic infection. Now, let's see, we have identified at least some differences, but now let's see what are the differences and try to understand, the avidity. Here, again, what I have already shown you before, you have a different concentration of your peptide, of the antigen that you use to induce stimulation of CD8 T cells as measured by interferon gamma production by flow cytometry. And then here you see just 2 examples of a response against an HLA-A restricted epitope from a patient from primary infection at baseline prior treatment that has a lower avidity as compared to this patient, which is a response from a patient with chronic infection, always HLA restricted. Now, if you see now, if we compare now in our 2 cohorts, the level of functional avidity in patients with primary infection as compared to patients with chronic infection prior therapy, and these are then therapy is not interfering with this measure, you can clearly see that patients with primary infection have a much lower functional avidity as compared to patients with chronic infection. Now, as you will see, this is a little bit different from what it has been published, because there was a paper, then we will go later, we will see, published in 2007 or 2008, that was saying exactly the opposite. It was saying that during primary infection, you have CD8 T cells with high affinity, and then progressing through chronic infection, then you lose cells with higher affinity, and then you have cells with lower affinity, which definitely is not supported by these data, which exactly say exactly the opposite. Now, the difference, as I said, is that in that case, there were few patients tested, they were not so early, probably during the recognition of primary infection. In contrast, as I showed you before, these patients are recognized within 2 weeks from the beginning of symptoms, very high viral load, average 27 million, then we really get these cells very, very early during primary infection. And then it's interesting also to see that CMV and EBV, they have very similar, the EBV, CMV, they have higher avidity as compared to CD8 cells from primary infection, and the same is for the flu-specific. There is a tendency, but there is no statistical significance. Now, one possibility, obviously, you could say, well, you know, the reason why we are saying that in primary infection we have lower avidity is because they are recognizing different epitopes. Probably it's due to the fact that you know, cells recognize different epitopes, they have different avidity. But if you look here, then we have looked at the common epitopes between a patient with primary infection and patient with chronic infection, and then you can see again that even with common epitopes, the patient with chronic, they have higher avidity as compared to primary infection. Then we went on with this type of analysis, and then we have seen what is the evolution of this response. These were primary infection at baseline, and then 1 year after treatment, a chronic infection at baseline prior treatment, and then after 1 year of treatment, and then this is a group of LTMP. Now, what is interesting here is the long-term non-progressor, okay, have exactly the same level of avidity that people with chronic progressive disease that are under treatment, that we don't see any difference. Then what the advantage that has been you know, suggested with regard that the LTMP will have cells with higher avidity as compared to chronic progressor, we are not able to compare. Then again, I think that the previous studies, they have really focused on one response. And then it's very hard to make really conclusion when you study one response in one patient. Anyway, the bottom line is that we confirm here, we don't see much evolution of the avidity under treatment. And then second, we don't see any difference between long-term non-progressor and chronic progressor. And again, long-term non-progressor have a higher affinity as compared to primary infection. Now, here I'm going to make the comparison again. These to me are sort of controls in order to see what are the, you know, just to make sure that these data that I'm going to show you really reflect, and then we always use as internal control the common epitopes, just to show you that again, we don't see any change. If we treat the patient, then the avidity again of CD8 cells directed against certain response remains stable over time if you treat this patient. And then if you look at, in contrast, as I said, if you compare also after 1 year of treatment, the difference between primary infection and chronic progressor remains significant for common epitopes. The same is with regard to LTMP, and then in contrast, if you compare common epitopes between chronic progressor and then LTMP, you don't see, you know, much difference. Again, I think these are internal control with strongly, you know, solidify, you know, these analyses that we have performed at the population level. Now, this was the paper that I mentioned to you before that was saying that there was a selective depletion of high affinity. And then indeed what we did here in our cohort, then we have compared, here are the frequency between lost epitopes, okay, lost responses, and those that are maintained, and that indeed what we see that the response that are maintained, they have higher frequency of CD8 cells. But if you look at the avidity between the responses that have been lost over time and those that have been maintained, we don't see any difference. Then there is not this takeover of cells with lower avidity as compared to the one that are eliminated earlier. Then I think this is another control indicating that you know, we don't see these early elimination of cells with high avidity. Now, we are going now to try to understand better why during, at the beginning of the infection, we have cells with lower avidity, and then why in chronic infection these cells, in contrast, acquire higher avidity. What is driving this, evolution? Now, okay, this is the conclusion from this first part of the studies. And then it gives me the opportunity to introduce, before moving to other data, to introduce a few words about the regulatory receptor, what regulatory receptor are. I think that there was a very large number of studies that have been published that on these regulatory receptor, and particularly the group of Rafi Ahmed, have shown that the expression of these receptors really have a major play in regulating the immune response, and then sometimes they are involved in the dysfunction that is associated with the immune response. And then, for example, the most well-known is PD-1, that was the first inhibitory receptor associated to immune exhaustion and increased susceptibility to apoptosis. And then, as you know, it has been identified as a marker of exhausted T cells in HIV infection. And then you can, after blockade of PD-1, to recover some function. And then, now, if we now look at the PD-1 expression, these are two examples, and then I'm going to show you the cumulative data. But here, this is a patient with primary HIV infection. At baseline prior treatment, this is a patient with chronic infection. We can clearly see the three negative receptors that we are looking at are PD-1, 2B4, and CD160. And then you can clearly see that there is a substantial proportion of cells that are PD-1 positive in chronic infection, but they are not at all PD-1 positive during primary infection CD8 cells. Then there is no evidence for exhaustion of CD8 cells during the primary immune response. And then CD160 is co-expressed by PD-1, with PD-1, obviously, again, no PD-1, no also CD160. And then here, if you look at this curve here, this green is going to give you the proportion of PD-1 positive cells in patients with chronic infection. It's about almost 50% of HIV-specific CD8 cells express PD-1 that are potentially exhausted. In contrast, during the primary infection, we have no more than 20%, maybe 10% of cells that express PD-1. Now, if you look now at the expression of these regulatory molecules in the different groups that I have shown you before, what it is clear that if you see, at 1 year after treatment, fundamentally you don't have any more PD-1 expression. Now, in chronic untreated patients, you have, as I said, almost 50% that express PD-1. If you treat a patient with chronic infection, you can reduce substantially the number of PD-1 positive cells, which this is a problem because, as you probably know, yesterday you have heard to talk about HIV functional cure. One of the potential treatment in order to rescue HIV-specific CD8 cell function, but at the same time to release, to activate CD4 cells infected is the use of anti-PD-1 antibodies. But as you can see here, PD-1 expression is driven by the presence of the virus. Then if you don't have the virus, you don't have PD-1. Then if you are going to treat patients that are controlled with virus replication, then you don't have the target. And then you may be completely useless to treat the patient with anti-PD-1. Anyway, and then you see here that long-term non-progressors have indeed the same level of PD-1 expression as a patient with chronic infection, but that have received antiviral treatment. Now, if we look now here, we are comparing here now PD-1 expression with the EC50, and then you can clearly see that there is a very nice correlation between the expression of PD-1 and your avidity. Then higher is the avidity, higher is PD-1 expression. Then fundamentally, if you have higher avidity cells, they have a tendency to exhaust much more than the cells that have lower avidity. And then it's the same here if you look at the cells that express all the negative regulators, PD-1, CD160, A2B4. Again, there is a very nice correlation. More cells have high avidity, more cells express these markers of exhaustion. Then fundamentally what it's telling you is that, as we will see also later, is that fundamentally what is driving this avidity, it's really the levels of virus replication. Then and then the virus replication is inducing exhaustion of your HIV-specific CD8 cells. Now, these are just again looking now at the comparison between the markers of differentiation, okay, in the different cohorts. And then again, what is telling you is that if you treat with therapy, you can revert the effector phenotype versus a central memory phenotype, both in primary, then in chronic infection, and then the long-term non-progressor have more a central phenotype, even in the absence of treatment. Then treatment is having an effect, and then what is this telling you again? That there is an inverse correlation between differentiation stage and avidity. Then if the cells have low, the cells, more the cells are differentiated, more you have higher avidity, and then vice versa. If the cells have earlier stage of differentiation, then there is low avidity. And then this is now with the co-stimulatory molecules is exactly the same. Again, we find, again, if you look at CD28, 27 expression, there is an inverse correlation, more expression of this marker, you have lower avidity. And then in contrast, if this marker are not expressed, effector phenotype, you have higher avidity. Now, this, I think that we have the opportunity in this cohort of patients with primary infection, then to monitor this patient for several years, okay? Then here you can see these are patients that have been followed for 5 years at least, and then some patients have remained under therapy, and then there is continuous effective control of virus replication, whereas other patients, they have spontaneously interrupt therapy, okay? And then it's going to be now interesting to see what are the changes that we observe in the phenotype, in the function, and in the avidity over time. Now, first of all, if you look here, this is a patient. This was before stopping therapy. The most important things that I want to underscore is that you can see here, this is IL-2. He had the IL-2 positive cells. After stopping therapy, this arrow means that in orange is when we stop therapy. The IL-2 positive cells have disappeared. Here there were few interferon gamma cells, and then when therapy is interrupted, a virus is back, there are a lot of single interferon gamma cells. Now, if you compare with the patient, in contrast, that has maintained therapy, has maintained virus suppression, then you don't see any change in the functional profile, over time. Then again, indicating that what is driving everything here, the changes are due to the virus. Now, if you look now, these are then the functional changes that you can see more clearly here. The patient with at 1 year of treatment, and then after the interruption of therapy, you can clearly see that there is a substantial change in the more single interferon gamma cells, whereas patients that remain therapy for a longer time, this is 1 year, this is 5 years, the same patient, you can see that progressively there is a much more polyfunctional phenotype. And now, this also just to tell you that why we think that everything is driven by the virus, this is PD-1 expression. Then if you see here, this is in the patient that has stopped therapy. These are the same response. These two patients are recognized the same epitopes. if the patient stop therapy, then you have expression of PD-1 coming out. In this patient that remains stable, then PD-1 does not express on these cells. And clearly indicating that what is driving also the exhaustion and the expression of these negative markers, it's really the presence of the virus. And then if you look then at the PD-1 expression, then you can see that patients that have that remain under therapy remain with very low levels. Then after treatment interruption, they increase the expression of PD-1. And then now, if you look at the PD-1 expression versus the percentage of single interferon gamma cells, then when you have effector phenotype, you can clearly see that there is a very high correlation. More you have the monofunctional interferon gamma secreting cells, more PD-1 expression you have. In contrast, if you have interferon gamma IL-2 secreting cells, double, then you see that there is an inverse correlation. Now, I'm going to show now in this patient what happens to the avidity. Now, this is a patient that has stopped therapy, and then you can clearly see this is the same response. This was when the patient was controlled. Then there was a low avidity response. Then the same response after treatment interruption, there is a substantial increase in the avidity. And then, in contrast, the same response, but in the patient that has remained completely controlled by therapy, you don't see any change over time. Then this is clearly, these are really, it's 40 weeks and 5 years later. Then, I mean, it's really a long time. And then you don't see any change here. Here, in contrast, is again 65 weeks and then almost 5 years. Then you see when there is interruption of treatment, then at this point there is a huge increase in the avidity. And here is the cumulative data of, we had the 12 patients that have interrupted therapy. And then you can clearly see again that we see, after treatment interruption, we see that there is an increase in the avidity. And then with the statistician, we did a mathematical model. And then what this model is telling us is that any time that you stop therapy, you have about 1 log increase in EC50. And then over time, even in a patient that is in the absence of therapy, you don't see much more. The slopes of the patient that are controlled and the slopes of the patient that are in the absence of therapy, they have the same, are very similar. Then the big difference in the big changes in avidity are only when you stop therapy. Then these results demonstrate that HIV replication drives the selection of HIV-specific CD8 T cells with higher functional avidity. These CD8 T cells have an effector phenotypic and functional profile and higher levels of exhaustion markers. Now, but this is not telling us what is the mechanism then underlying these, why these cells increase avidity. Now, there is a fundamental difference. B cells, they increase avidity for the antibody through hypersomatic mutation. Then this mechanism cannot apply to T cells because does not exist a hypersomatic mutation for T cells. Then there has to be different mechanisms. Then how then really we can explain that against the same response, against the same epitope, then a T cell is able, depending from the presence of the absence of antigens, to change the avidity. But also what it's telling us is that you don't have for a fixed, any given response, you don't have only a fixed level of the avidity, but you can change over time depending on the condition of antigen exposure. Then what we did, we took advantage of a large study that we performed that was published last year, that we look at TCR diversity, just to tell you what was the strategy that we have used. Then we isolate HIV-specific T cells. These T cells are tetramer positive, they are sorted, and then we make a V-beta TCR amplification. And then we make, we look at CDR3 length by spectral typing, and then we make also, deep sequencing. And then what we show in this paper, which was something quite unexpected, is that generally, the level of diversity for any specific response was generally was thought to be that generally you have about 9 different CDR3 recognizing the same epitope. Then what we have shown in this paper that the average number of CDR3 that recognize one epitope is about 40. Then it is a huge, TCR diversity. Anyway, we have used these strategies. Then I'm going to show you the data. And then what here we show, these are, if you look at now, these are the changes here. We are looking at percentage of CDR3 renewal. Then we calculate, what are the changes that are observed in the CDR3 under treatment or after treatment interruption. Then you can see that there is a substantial increase in TCR renewal. And then what we can show here, that there is a nice correlation between the fold increase in the magnitude of the response and also the level of CDR3 renewal. And then the most important is that there is a correlation between fold increase in functional avidity that we see in CD8 T cells and then the percentage of CD3 renewal. Then the conclusion is that virus exposure leads to the selection of high avidity HIV-specific CD8 T cells through an increase in TCR renewal. Then fundamentally, in order to increase avidity, you are going to recruit new clones of CD8 T cells, and then eventually, in a very stochastic process, then you select for cells which eventually have higher avidity. And then at the same time, these cells, as now we are starting to have some evidence, they exert selective pressure because in collaboration with Jim Mullins, we are performing a very large study. Then we can see that now the cells with higher avidity, they induce some mutation in certain points in the HIV genome. Now, these are the people that particularly is a PhD student that has made all these works, Elena Vigano, in my lab. And then before, if I have 2 minutes, I want to just to show you 3 slides, which are very interesting with what also Mattia has been talking yesterday about HIV functional cure, which I think that there is a very good news about what I'm going to show, but there is also some, some thoughts that is driving. Now, this is a patient. It's part of this cohort of patients that this patient we have identified 2 weeks, not after the start of symptoms, 2 weeks after he was exposed to HIV. And at the time of primary infection, he had 25 million copies. And then you can see this patient was put immediately, within 48 hours, under treatment. And then the treatment was, as usual, very effective. And then spontaneously, the patient, 1.5 years later, almost 2 years, then he decided to stop therapy. Okay? Now, the patient stopped therapy. And then for 10 years, because here we have in 2000, and then for 10 years, there was a little blip 2 months after he stopped therapy, was about 67 copies. But then for 10 years, the patient did not have any virus. We couldn't find any virus. We have done several times leukapheresis. We sent the cells to Theo Work, at NIH. He was unable to isolate virus. He has found the lowest DNA proviral copies per million of CD4 T cell. gut biopsies. No way, there was nothing. And then look at also CD4 cell counts, because we are talking about in the range of 2,000 CD4 cell counts. Now, the problem is that this patient then in 2010 was still below 100 copies, okay? He had a little bit about 66 copies here. And then in 2011 is 372 copies. And now we have a point a few weeks ago, and then it's 270. Then what is remarkable in my mind is that the patient now, after 10 years that was not, we could detect virus, now it's 1 year and more that is controlling, his own virus. Now we know already that it's not superinfection. We have done this with Jim Mullins, that we know that it's the same virus, but we are finding very interesting mutation, evidence for some escapes. But the patient, why I think that is interesting, because it does not have any of the genetics associated with protection. And this guy is not TCR5 wild type, is not B27, is not B57, and it's really a very clean patient. Now, something that he has unique, this patient, and that to me gives some hope also for the T-cell control, is that this guy had a very unique functional profile, because we have never seen that 80% of the cells, they are able to secrete IL-2. I'm not going to show you the data, I don't have time, but he has these cells proliferate very well. Then all these cells have, at least they had, when he was controlling, a central memory phenotype. Now, what I think that is interesting, and then I think that we should pay much more attention and give value when we see changes in immune response in the functional profile, because this was his profile in 2007, when he was not controlling. Then in 2009, he was still undetectable, but we saw that there was a change. There were many less polyfunctional cells, okay? And then 1 year later, when he has made his blip at 66 copy, you see that fundamentally he has lost a lot of polyfunctional cells. Then it seems that this is a very sensitive measure to see what is the virus activity, because over time we have seen this change, and then even we have seen a reduction 1 year earlier of what we have been able to measure in the plasma. The other thing that is unique, and then I think that is a little bit sad, if we think about a vaccine or T-cell vaccine that can induce, can control. Now, this is now the number of responses that this guy has. After, even if it is controlling very well, he has developed many more responses. But what I think that is amazing, that is that 20% of these CD8, total CD8 cells, are HIV-specific. Then considering that he has 2,000 CD8 T cells per microliter of blood, then he has about 400 CD8 T cells circulating, which are HIV-specific. Which is really huge number. Then I think that there is we feel with Jim Mullins that we have evidence here that really there is a CD8 really mediated control, also because this guy does not have antibody. We cannot isolate any neutralizing antibody. Then the response seems to be T-cell mediated, but I don't think that we will ever able to induce with the vaccine these, first of all, this number of responses, but in addition, this quantity of CD8 T cells. Okay, we'll stop here. Thank you for your attention.